Good morning. My name is Angela Wood. I'm from the University of Cambridge, and I am delighted to start this next session, both by I'll be chairing this session and also presenting a background in this session, and I'm going to present to you a background of the CVD-COVID-UK consortium. Now, the CVD-COVID-UK, as you've already heard, we are aiming to understand the relationships between COVID-19 and cardiovascular diseases through the analysis of linked, nationally collated and healthcare datasets. And to achieve this ambition, the British Heart Foundation Data Science Centre has developed a UK-wide infrastructure where approved researchers have efficient access to patient-level healthcare datasets through secure and trusted research environments across the 4 nations shown here. Now, such data and environments have been available for Wales and Scotland and accessed for research use for several years. But at the start of the COVID pandemic, researchers had no access to national linked healthcare data across the population of England. And so to address this gap, CVD-COVID-UK established a novel population-wide resource in partnership with NHS Digital. As you can see on this slide, the resource comprises of a range of linked datasets covering the entire population of England, including hospital data, death registrations, primary care data, dispensing and e-prescribing data, COVID-19 vaccination and lab test data, maternity and mental health data, and data from intensive care units and from cardiovascular specialist registries. And these linked person-level records are accessible through the new NHS Digital's trusted research environment for England. And this environment ensures a secure storage of and access to the data, but minimises any security risks of data travel. So researchers go into the trusted research environment to access and analyse the data. And the resource includes detailed information on over 55 million people, and that's more than 95% of the population in England, providing a statistically powerful resource. There is comprehensive information on characteristics and health outcomes, and in particular, there's around 95% complete data on key characteristics of the individuals, such as age, sex, and ethnicity, as well as information on geographical locations, socioeconomic, health, and personal characteristics. And all these datasets are kept up to date on a monthly basis. So currently, we have population-wide coverage of data from England, with a population of over 55 million, as I've said, but also from Scotland, and this is through the Safe Haven, with a population of over 5 million, and from Wales through the Sale Data Bank, with a population of over 3 million. And work is underway to extend further to Northern Ireland. The CVD-COVID-UK consortium has UK-wide membership of over 250 interdisciplinary members from over 40 NHS and academic organisations. The consortium supports over 70 approved analysts across the different trusted research environments, and together we're addressing over 30 approved projects, with many more being approved at the moment and on their way. Now, we advocate best practices for open science, and all protocols, statistical analysis plans and computer algorithms have open access documentation available on GitHub, and we have a growing number of published outputs which you can view on the web pages, and also you're going to hear about in the following talks as well. So to access these resources, you'll be interested to hear the process. So researchers must first have a proposal which is approved by the Approvals and Oversight Board Committee. So this process includes a meeting where the project leads and members of the patients and public panel meet to discuss the relevance of the project, the importance of the proposal, as well as ways to involve and engage patients and the public through the course of the project. The board then decides whether each project is approved with conditions, or not approved. And this whole current time period from submission of a proposal to approval is around 1 month. And the BHF data science team are looking at ways to reduce this already impressive time frame. And here is a summary of some of the projects that have been approved, ranging from projects centred on methods, such as high throughput phenotyping approaches, projects focused on medicine, such as assessing COVID-19 through medicines, and a range of others, including the impact of COVID-19 infection and vaccination on vascular health. And actually, we'll be hearing about these 3 projects in the following presentations. Now, you'll also see on this list that there's some projects that don't look like they are to do with cardiovascular disease. And actually, in order to address a wider range of policy relevant questions for COVID-19, the BHF Data Science Centre recently successfully got the approval to broaden the coverage from research on cardiovascular disease to all disease areas. So you can see projects such as the impact of COVID-19 on eye diseases on this list. And also you'll see the renaming of the consortium and the title from CVD-COVID-UK to COVID Impact. So I'm now just going to show you how linking data from the different healthcare settings is essential for ascertaining disease outcomes. And here I'm showing it for cardiovascular disease events. And in particular, looking at stroke and myocardial infarction here. So from around 55 million people, we identified nearly 100,000 people with a first ever stroke or transient ischemic attack. And these are data from January to October 2020. Importantly, from this, you can see that the data comes from the different resources, from primary care, hospital admissions or death registrations. And around 30% came only from primary care data and 4% only from the death registry records. Similarly, we see something similar for myocardial infarction, where we identified nearly 64,000 events, and 8% of these were recorded only in primary care and 12% only in death registry records. Likewise, linking data from different healthcare settings is essential for providing a full picture of COVID-19 infections across the population too. We've identified 3.5 million people with COVID-19 by mid-February 2021, concording well with the Public Health England official statistics. 3.1 million of these were identified as people with a positive test, whilst 2.4% were diagnosed in a primary care setting. And you can see that there's around 8 to 9% which were only reported in the primary care setting. And this is especially important due to the testing procedures which were not widely available early on and now later on in the pandemic. And we identified 364,000 hospitalised COVID-19 infections, which accounts for around 10% of all infections. And we'll see further detail on this work by the talk by Chris Tomlinson coming up. So to summarise, the CVD-COVID-UK has made it possible for researchers to conduct research at this population-wide scale through the establishment of NHS Digital's new trusted research environment for England. It's enabled many well-powered studies and insights that we're about to hear about. But finally, I know that this has been said before, but this has only been made possible by having a national coordination and a multidisciplinary team science approach, as well as the public support and trust. And with that final note, I'm now going to hand over to Doctors Tom Bolton and John Nolan, who are the Senior Health Data Scientists at the BHF Data Science Centre. And they're going to provide to you an overview of how, well, basically they're making life easier for researchers by clearing a pathway so that researchers can come and make efficient use of the resource that I've just described. So Tom and John, I've got a great pleasure of handing over to you.
Thank you very much, Angela. Hi, everybody. So as Angela has just said, my name is John Nolan. Tom Bolton and I are both Senior Health Data Scientists with the BHF Data Science Centre. We will be talking today about the work we are doing to hopefully help make life easier for researchers and to speed up the pathway to research-ready data. So once a researcher has had a project approved and has gained access to the TRE, what are the main challenges that they will face? Well, first of all, the DSC holds a huge amount of data, over 60 datasets across 3, soon to be 4 TREs. Most projects require multiple datasets to be brought together to answer a research question. Some of these datasets may be familiar to researchers, but others almost certainly will not. These datasets will all need some level of cleaning and exploratory data analysis before they can be used. This work can be a very time-consuming aspect of any research project. We also have datasets with very large volumes of records. So in the examples here, 7 billion primary care records in England and over 4 billion prescribing records. This brings us on to the second challenge. Many researchers will have expertise coding in their analytical software of choice, frequently RStudio or Stata, but not all will have coded in PySpark, Python, or SQL before, and they will need to learn these in order to be able to bring together these large volume datasets to create their final analysis dataset. Finally, for those analysts carrying out their project across more than one TRE, there are differences in the data across the 3 nations and TREs. For example, primary care differs in both its format and scope in the 3 TREs. This can make it difficult to replicate research projects exactly across the TREs. Trusted research environments also contain different software packages, and so researchers may need to write their data curation pipelines in different languages. So what are we doing to help researchers? When working with datasets in the TRE, researchers need to spend a lot of time curating and cleaning datasets before using them. As an example here, we have some synthetic hospital episode and death data. We can see that the patient ID name differs in both datasets. Dates are in different formats and may even be stored as strings. The death records contain duplicates, which will need to be removed before joining to other data sources. And the diagnostic coding in the hospital data is in wide format over multiple fields, which will all need to be checked for code matches. We aim to build a curated layer of data in the NHS Digital TRE that takes care of some of these initial cleaning steps and removes the need for researchers on different projects to repeat these same tasks. This includes, but is not limited to, standardizing variable names and formats across datasets, removing duplicate rows, and reshaping data in order to make it simpler to match code lists to get outcomes, covariates, etc. for the research cohort. We also want to help researchers to hit the ground running by helping them to understand all of the data sets that they are going to be working with. So for each data set, we aim to create documentation that summarizes the data and provides the key insights that they will need to be aware of. So we've started developing PySpark notebooks for each data set in the NHS Digital TRE that will provide this. By notebooks here, we mean a coding document that contains runnable code and data outputs and visualizations alongside narrative text. These notebooks will contain dataset summaries, so things like batch checks to make sure the data has been updated correctly and that record counts make sense. Data coverage plots to help researchers know when data is complete too, so they can identify a censor date for their study. But more than this, we want these summaries to identify and highlight what we think are the key findings that researchers need to be aware of for each dataset. And I'll provide a quick example of this on the next slide. We want these notebooks to be a useful coding resource, particularly for new analysts just joining the TRE. Researchers will be able to see PySpark code for connecting to database tables, selecting fields, grouping, joining, and summarizing data, and code for producing visualizations. We envisage that these notebooks will be available for all TRE users in a shared workspace, that they will be refreshed after every batch update and continually updated with new analysis and insights, also with input from our researchers. Finally, just to give an example of the type of insight that we want these notebooks to provide, when exploring the HES critical care data, we could see that the number of patients and records increased dramatically from April 2017 onwards. This confused us until we looked at the data split by age group, and we could see that the increase was driven entirely by patients aged under 18. It turns out that from 2017 onwards, the critical care data started including neonatal and paediatric admissions, something we weren't initially aware of. And this is an example of the type of finding that we would want to flag up to any researchers about start using the critical care data. I'll now hand over to Tom, who will talk about the development work we're doing on data curation pipelines.
Thank you very much, John. So what do we mean by a data curation pipeline? Put simply, it's the process of transforming the collection of raw or original datasets to a research-ready analysis dataset that can be used to answer research questions. It can be one of the most time-consuming parts of a project and will typically include data management, wrangling, cleaning, harmonization, phenotyping, checking, and visualizations. Working across the three trusted research environments where the underlying data differs, the aim is to make the data curation pipelines as similar as possible to produce consistent research-ready datasets to allow the same or almost the same analytical code to be run on each dataset. Broadly speaking, a data curation pipeline might have the following stages: initial data management, wrangling and cleaning to produce curated snapshots of raw data, cohort selection, applying inclusion-exclusion criteria, extracting key patient characteristics such as age, sex, ethnicity using multiple harmonized data sources, performing quality assurance to remove erroneous or suspect records, creating code lists, and using these to create covariates, outcomes, and exposures. And this was the pipeline used to prepare the research-ready analysis datasets for our studies investigating the risk of thrombotic events following both COVID-19 vaccination and infection. And all of this code is available via the BHF Data Science Centre GitHub repository and has formed the basis for the pipelines that John and I are now developing. Today, I'd briefly like to showcase some of our work around code lists, covariates, and outcomes, where we've been generalizing the pipeline to be readily applicable to multiple projects. So one of the most important stages of the data curation pipeline is constructing the code lists. So here we have the first few dozen rows summarizing the number of codes in our code list by name and terminology. The table actually contains around 60 names, more than 13,000 codes across 4 terminologies, containing a mixture of existing and novel code lists. Focusing on the first row, acute myocardial infarction, AMI, we have 80 codes, 5 ICD-10 codes, and 75 SNOMED CT codes. And here we can see the 5 ICD-10 codes and the first 20 of the 75 SNOMED codes based on caliber and published in the HDR UK phenotype library. And John and I have included data checks, validation, and reformatting to ensure accuracy in the code list construction process. So once we've defined our code list, we can then apply it to create covariates for our cohort. So taking the study population of around 45 million individuals from England that we identified for the COVID-19 infection project, and focusing on GP records from the GDPR table and hospital admissions from the HES admitted patient care table, we can filter records prior to our study start date of the 1st of January 2020 and join these to our code lists. Continuing with the AMI example, we find almost 1.2 million individuals have records with codes matching our code list, and we can see how this varies by data source, and we can look at how the codes matches by individual codes as well. We can also identify an individual's first or earliest matching code, and we can see how these first events are distributed over time, looking at the number of individuals per year. We can look at this by data source and by the codes with the highest frequency for each data source. We can also look at the distribution of key patient characteristics, such as sex and age. And John and I have developed code to automatically produce these summaries and visualizations for every covariate, which provides a greater level of transparency and understanding when curating the data. We can apply exactly the same methodology to generate outcomes. This time we're also including the deaths table as the source and are looking forwards in time up to the study centre date. Continuing with the AMI example, where certain covariate-only codes have now been excluded, we find 130,000 individuals with an event in the follow-up period. And we've generated the data source and code summaries automatically, and the distributions over time, looking at the number of individuals per week here by data source, codes within these, and patient characteristics. This work is all aimed at making life easier for researchers and accelerating research by developing the processes and tools that can be readily applied to new projects. I'd like to just end our talk by thanking those who contributed to this work. John and I will be presenting a deeper dive at the monthly webinar on the 27th of April, which will be available on the Centre's YouTube channel shortly afterwards. Thank you very much for listening.
Thank you very much, John and Tom, for describing all the absolutely essential work that you're doing for us in the different TREs, and it's really greatly appreciated. And as I said, really speeds up the process for analysts to get their job done. So we now have a series of 3 scientific talks, 5 minutes each. It's my understanding that Chris is next, but I can't see him on the call. Is that correct? Okay, thank you. So we're going to go straight to Venexia instead. Venexia is a research fellow in medical statistics and health data science from the University of Bristol. And she's going to present her work to us today on the association between COVID-19 infection and vaccination and different vascular diseases using the resource that we've just described. So over to you, Venexia.
So the first of these, the paper on the left, looked to quantify the association between time since diagnosis of COVID and vascular disease. And that's available as a preprint from medRxiv. And then the paper on the right looked to quantify the association of vaccination with either the Oxford AstraZeneca vaccine or the Pfizer vaccine with major arterial, venous and thrombocytopenic events. And that's available from PLOS Medicine. I'm presenting these papers together because they share their methodology. And that is that both used a cohort design with a Cox proportional hazard model to estimate hazard ratios with time since exposure. And it was critical for this analysis to account for changes in rates of outcome events with calendar time, which we know have varied during the pandemic. So for that reason, we set time 0 in these analyses. So for the infection paper, that was set to the 1st of January 2020, which is approximately when the pandemic started. And then for the vaccination paper, that was the 8th of December 2020, which is when the UK vaccination programme started. In order for these models to run, we had to be mindful of computational efficiency. So for this, we used a sampling approach where we included all individuals with the outcome and a 10% random sample of those without the outcome. We then used inverse probability weighting to recover the full population. So moving swiftly on to some results, starting with the infection paper. Here you can see results for 8 different outcomes. And in black and grey, we have the extensive and age, sex, region adjusted. And then in the red colours, we have the hospitalised versus non-hospitalised. And you can see across all these different outcomes, they follow the same general pattern where we have very elevated risks in the first couple of weeks following COVID infection that attenuate over time with weeks since diagnosis. But in a lot of cases, these don't get back to a hazard ratio of 1. Focusing in on a combined outcome of any venous event, we can look at particular subgroups in this analysis. So the top left and top center panel reflect the last graph, but in the top right, we have prior history of event. We're also able to look at age group, sex and ethnicity. So some notable things here, we see that people that are younger had lower hazard ratios and the non-white ethnic group seem to have higher hazard ratios compared to the white ethnic group, but relatively little differences by sex. Moving now to the vaccination results, I'll focus just on 2 outcomes that were highlighted as potential adverse events for vaccination, starting with intracranial venous thrombosis. So here in the under 70s, we saw an elevated hazard ratios following AstraZeneca vaccination, but not for Pfizer. And in the over 70s, the hazard ratios were similar to 1 throughout. And then for thrombocytopenia, we saw relatively little difference between AstraZeneca and Pfizer, and hazard ratios close to 1 throughout for people under 70. And for the over 70s, we saw a suggestion of a protective effect, but this probably relates to the fact that vaccination is reducing infection. Thank you for listening, and thank you to all of those that contributed to these papers and are listed on this slide.
Thank you very much, Venexia. And we're going to move on to our next talk, but before I do, I can see some questions are coming in through the question and chat box, so please do keep putting those in and we will come to them at the end. I'm now going to hand over to our next speaker, Dr. Chris Tomlinson, who's going to present to us some work on phenotyping COVID-19. And Chris, when you start your talk, you'll just have to go backwards a few slides because they've gone out of sync, but I'll hand over to you now.
Yep, apologies for that, just rewinding through time now. Okay, good morning everyone. My name is Chris Tomlinson. I'm a anaesthetics and intensive care registrar and PhD student at the UCL Institute of Health Informatics. I'm going to talk to you today about our experience of phenotyping COVID-19 and insights from linked data for 56 million. So this is a brief overview of the COVID-19 phenotyping process, work carried out with my IHI colleagues, Dr. Johan Tusen and Spiros Danaxis, amongst many other consortium collaborators. We use 7 datasets spanning primary and secondary care with multiple terminologies and proprietary fields to define 10 COVID-19 phenotypes. These span positive tests, primary care diagnoses, hospitalizations, ventilatory support modalities, ICU admission, and death, reflecting disease severity and outcomes. For further details, I'd encourage you to read our preprint via the link and QR code. You've already seen this slide from Angela's presentation showing individuals identified from each group of datasets. Positive tests in dark green, primary care diagnoses in light green, hospitalizations in orange, and deaths in blue. From this, I'd like to highlight 2 take-home messages. Firstly, linkage maximizes ascertainment. We identify unique cases from each dataset, and in this non-proportional Venn diagram, there are significant numbers in each cell. Secondly, linkage allows us to uncover important subgroups. For example, the light blue cell shows 10,500 individuals identified only from deaths data. And the blue set outside the orange set shows almost 40,000 individuals who died without any hospital contact. I'd now like to focus on a particular phenotype of interest, that of non-invasive ventilation. Again, we utilize linkage to maximize ascertainment by linking across 4 datasets using both OPCS4 procedural billing codes and proprietary fields. In this very simplified representation, you can see the relationship between 3 of our COVID-19 phenotypes: hospitalization in red, non-invasive ventilation in orange, and intensive care admission in blue. Here, linkage allows us to identify the 22,000 patients who received NIV outside of intensive care, and that represented 40% of all patients treated with NIV. We believe we're the first to demonstrate this at such scale, and that is only possible through linked national data. In summary, I hope I've demonstrated that linked data on a population scale enables comprehensive phenotyping of COVID-19, maximizes case ascertainment, and allows identification of key patient subgroups that would otherwise fall through the cracks. For further details, I'd encourage you to read our preprint. Thank you very much for listening, and to all those who made this work possible.
Super presentation. Thank you very much, Chris. And again, I'll encourage you to keep putting your questions into the chat function, and we're going to move on to our final talk in this section from Dr. Caroline Dale, who's a Senior Research Fellow at UCL, and she'll be presenting the work her and her team have been doing on using medicines to understand the impact of COVID-19. So welcome, Caroline, and over to you.
Thank you, Angela, and thank you very much for giving me the opportunity to present this work on behalf of our medicines group. So the direct impact of the COVID-19 pandemic is well established, but the indirect impact has been less well studied. And so in this research, we'd use medicines to measure the impact of the pandemic on the management and prevention of cardiovascular disease. So we use medicines as a proxy for cardiovascular disease risk factors such as blood pressure, lipids, and type 2 diabetes. And we did this by categorizing each cardiovascular drug substance according to its primary cardiovascular risk factor of use. And after that, we were able to quantify the implications of non-treatment with the medications for future cardiovascular disease events. So this enabled us to analyse trends both in the overall pattern of cardiovascular disease medications, as we see on the left-hand figure, and the patterns by individual cardiovascular disease risk factors. So looking first at the left-hand chart, we can see that ahead of the first national lockdown, we see a clear uplift in the prescription of CVD medications, which is similar to the pattern that we commonly see ahead of Christmas every year. Now, when we consider that same uplift on the right-hand figure, you can see that that uplift differed according to CVD risk factor, and that some CVD risk factors, in particular, we can see the indicated by the green line, insulin, saw even more marked increases. And for example, insulin was up 24% in March 2020 compared to the previous year. Furthermore, if we look at the right-hand side of that graph, after the first vertical dotted line, you can see that most CVD risk factors show percentage year-on-year change below zero for many months during 2020 and into 2021, indicating that the management of CVD through medication was lower after the onset of the pandemic. We also looked at trends in counts of incident medications. And by incident medications, we mean that the first time a person was given a medication within a particular CVD risk factor subgroup. So, for example, looking at the top red line, this is the antihypertensive group, and you can see that there was a marked drop immediately following the first national lockdown. And comparing pre-pandemic trends to after the first national lockdown, we estimate that approximately 24,000 people less every month are taking antihypertensive treatments for the first time than had those pre-pandemic trends continued. And accumulating that across the study period, we found that almost 500,000 people were missed to have been commenced on antihypertensive treatment through the period of March 2020 to May 2021. Now, because the impact of cardiovascular medications on cardiovascular events is well established from previous research from clinical trials, we were able to model what the effect of that missed medication would mean for future cardiovascular disease events. And so we estimate that missed treatment just of antihypertension alone would lead to a future additional 13,659 CVD events, including almost 3,500 strokes and almost 2,300 myocardial infarctions. So in this research, we have shown that a medicines lens can be used to complement existing research using diagnoses to deliver rapid understanding of ongoing trends in healthcare service provision and in the case of the COVID-19 pandemic, the reduced use of routine services. We found that the detection of CVD risk factors indicated by new initiators on CVD medications had not returned to pre-pandemic levels by the end of the study period, and almost 500,000 people had not initiated treatment with antihypertensives, which will contribute to increased future CVD events and add to the indirect costs of the pandemic. Finally, we think that this research helps to hint at the potential for linked national healthcare records to help deliver novel treatment routes, for example, perhaps through community pharmacies or the NHS app, that don't require face-to-face interaction with primary care providers, such as vascular health checks. Many thanks for listening to the presentation, and I'd like to acknowledge all the people and organisations that have contributed to this work. Thank you.
Thank you very much, Caroline, and thank you to all the speakers who have presented to us this morning. And now's a chance to have a bit of an open discussion about some of this work. There's a couple of questions that I can see in the chat, and I think we're going to first of all go to Venexia, if that's okay. Nicholas Mills said that was a nice presentation and great data. Maybe you could just describe a little bit more how you defined hospitalised and non-hospitalised COVID cases. In particular, Nicholas says that the risk of major adverse cardiac events seems higher than he would expect in those who are not hospitalised. So Venexia, do you want to respond to that?
Yes, thank you for the question. So hospitalisation for COVID-19 was defined as a admission record with confirmed COVID-19 in the primary position within 28 days of the first COVID-19 diagnosis. And then everyone without that was defined as without hospitalisation. That is written in the preprint, because I'm aware that me stating it like that, it probably isn't the easiest to remember. But yeah, within 28 days, primary position.
And Venexia, I wonder whether you want to comment on the risk of major adverse cardiac events seems higher than he would expect in those hospitalised. Sorry, in those who are not hospitalised.
Yeah, so this is interesting. There's a couple of interesting results like this. A bit like the prior history, people with prior history also had elevated hazard ratios compared to those without prior history of these events. And I think lots of it's to do with how we're ascertaining both the events and the COVID infection. So that we did a lot of work around things in primary positions and in other positions, and what came first in terms of the events and the infection.
Thanks, Venexia. And there's another question here from Tom Yates. Again, it's back to you, I'm afraid, Venexia. It's about, to what extent do you think the associations between COVID-19 and vascular events are causal versus just a measurement issue? In particular, Tom says that the people hospitalised with COVID were probably much more likely to have investigations, such as for pulmonary embolism, particularly over the period where healthcare usage for non-COVID conditions started to fall.
Yeah, so we tried to use as much of the data that we had access to as possible, and we gave a lot of consideration to the timing. So as I mentioned during the presentation, we anchored using calendar time to try and account for the fact that ascertainment of outcome events have changed over time during the pandemic. But of course, nothing's perfect and we can't rule out measurement error.
Yeah, no, thank you. I can't see any other questions in the chat. Maybe that's because I'm not viewing them properly, but actually I have a question for you, Caroline. And really, really interesting work to see the effect, to kind of understand COVID-19 through medicines. My question to you, and you might not know the answer, is, looking at the trends that you've seen, when do you think it will get back to normal with regards to medicines being dispensed at the pre-pandemic level? I know you showed the trends and we're certainly not back to normal yet, but have you done any kind of trajectories to kind of estimate that length of time?
No, thanks for the question, Angela. No, we haven't done any prediction work. I guess obviously we can update the study period now that we've got more data. But my understanding from talking to other colleagues is that it has not yet returned to the pre-pandemic levels. Obviously, there's a lot of comments on potential alternative explanations that might have led to those declines, which I didn't put into the presentation. So people have raised previously whether it's changes in the demographic structure of the UK, or was it due to obviously the additional mortality, people dying from COVID itself, that led to those declines. And we have done quite a lot of sensitivity analyses around that and showed that no, that's not the case, that we don't believe that it was those demographic changes that were leading to the declines. So, but to answer your first question, no, we haven't actually done any future projection work yet.
No, thank you very much. And then maybe I've got a really tricky question for Chris. Chris, if it's okay to ask you this, and that's really, what are researchers going to do now that people are not getting tested for COVID-19 or actually even seeking any healthcare if they believe they are COVID positive? What does it mean to researchers?
So yes, that's a challenging question for me, and certainly a challenging question for researchers too. Certainly our work in this phenotyping exercise has only been based on the sort of observed data and what we could measure. We've tried to mitigate for the lack of COVID testing in the early phases of the pandemic by using clinical diagnoses, particularly from primary care, and that did add to ascertainment. That becomes even more challenging going forward, especially if, through protective effects of vaccination and maybe milder disease, patients are even less likely to engage with healthcare as a whole, not necessarily just testing. And yeah, that's that certainly is tricky. I'm afraid I don't have a particular answer off the top of my head as to how to manage that. I think something for us all to think about going forward.
Yeah, no, and I just wanted to bring it up. It is going to be a challenge for us, isn't it, moving forward? And so I'm going to move to another question here. This is from someone with no name, but the question is, so I haven't read it out yet, so I don't know who it's probably for. Maybe Venexia. Let's wait and see. So, do you find evidence to suggest that incident events identified as non-overlapping across different datasets may be differentially associated with risk factors or other baseline characteristics than those occurring more commonly in the databases? So I'll just read that out again. So, is there evidence to suggest that incident events identified as non-overlapping across the different data sources may be differentially associated with risk factors or other baseline characteristics than those that you find from events from overlapping the different databases? I'm not sure our team have actually looked at this. Venexia, have you ever looked at it, or even Chris, kind of looking at the subsections, I guess, of the Venn diagrams to see whether we get different associations or associated with different risk factors?
Yeah, we certainly haven't looked at anything in relation to risk factors. I wonder if this is directed at actually Tom and John, who had looked at the different data sources. Because in our analyses, we take information from all of the data sources wherever possible. So we haven't focused on where there's non-overlapping data.
And John, Tom, I think you've explored this, but haven't taken it far enough to look at associations with different risk factors or other characteristics. Is that correct?
Yeah, it's not something we've looked at in any great detail. It's the type of analysis that I think we would plan to do when reviewing things like the NICOR data sets and looking at the overlap between NICOR and other sources to see if there's variations in the patterns we see of the patients that are identified in a single source or across multiple sources. But we've really just scratched the surface on that. But it is something I guess we would like to look at with a focus on particular data sets.
Super questions. Thank you for those who've submitted their questions. I think I'll wrap this session up now, thanking all of our speakers for their preparations and presentations this morning. And I'm pleased now to hand back over to Professor Cathy Sudlow, who is going to chair the next session. So thank you all.
Thanks very much indeed, Angela, and thanks to all the speakers. A really great, very rich, diverse session there. It gives me great pleasure to welcome our second keynote speaker of the day, Professor Deborah Lula, who's the British Heart Foundation Professor of Cardiovascular Science and Clinical Epidemiology at the University of Bristol. Her main areas of research are in life course and genetic epidemiology of cardiovascular disease, diabetes, and women's reproductive health. And she also has particular methodological expertise and interests in examining causality through a range of different approaches. She's had an enormously distinguished career, still going strong, but it includes being elected as a Fellow of the Faculty of Public Health by distinction in 2006, a Fellow of the Academy of Medical Sciences in 2012. She's been named by Thomson Reuters in multiple years as being in the top 1% of scientists who are most influential in their field. And she was also awarded a CBE in 2017 for her services to social and community medicine research. So, Deborah, thank you very much for joining us today. We're really delighted to have you and to hear you talk about South Asian white European differences in life course cardiometabolic risk. Thanks. Over to you.
Thanks a lot, Kathy, and thanks for inviting me and for all of the talks so far. They've been absolutely fantastic. This is a talk in which I will introduce the Born in Bradford cohort and show you some research results, because I think people are always interested in them. But in terms of this meeting, it is about how we have linked the cohort data to a whole load of other routine data, why that's important and what it might mean going forward in HDI UK more generally, and also in the centre. So by way of background, Bradford is often seen as a basket case, for want of a better word, and if you're born and brought up there as I was, it's frustrating because the sorts of stereotypes that are used to describe the city are not the reality. So this is just a picture of my mum when she was pregnant with me. She's in front of a car because they hadn't seen a car on the street before, and I was born in this house here. That's me and one of my sisters at secondary school. But it is a vibrant and exciting city with lots going on for it. It has, because of the mixed ethnic background, because of the deprivation in the city, it's one of the most deprived cities in the UK, it has a lot of health challenges. And the Born in Bradford cohort started really because going back to 2005, I had been working as a clinician in Bradford, but I'd moved to Bristol by then and started an academic job, but maintained strong links. Bradford had one, if not the highest perinatal and maternal mortality in the country. And so there was a large inquiry as to why that might be, and lots of different clinical audits. I was involved in an audit that looked at the extent to which we followed NICE guidelines in the diagnosis and management of gestational diabetes. And gestational diabetes is diagnosed in the UK and in most places by a diagnostic test at around 24 weeks of gestation. There's a screening at the first antenatal clinic based on risk factors as to who should get that diagnostic test. And based on just two of those risk factors, so women of South Asian ethnic origin and high body mass index, over 80% of the pregnant population in Bradford should have been referred for a diagnostic or a glucose tolerance test, but only 20% were. And when we talked to the midwives and the obstetricians, the overwhelming response was, so what are you telling us? All of the pregnant women in Bradford are unwell, are ill, are at risk, are abnormal. That doesn't make sense to us. And it was as if they were being overly selective in the sense that you don't consider pregnant women as being diseased and unwell. They're not patients in that sense. But what we then argued was, well, in these kinds of circumstances, the screening sort of doesn't make any sense. And really, we should just offer universal tests to everybody and say, this doesn't mean everybody's ill, but it does mean we should be testing everyone. And that was agreed by the local commissioning group. What then happened was John Wright, who's been very instrumental in Born in Bradford, said, Deborah, this is crazy. We should make this into a cohort. Let's do it. And that's where Born in Bradford came from. We had no core funding originally. And I think two things came from that. One was going back to Kelvin's talk and a theme that's gone through this. We absolutely had to work with the community. We had to get everybody involved in the design of the study, the setup of the study, why we were going to do it. And that's continued to be the situation, where we started in January working with the original children who are now starting secondary school to co-produce the next 7 years of follow-up. What are the things that they want us to study? How do we best do that? So it's challenging, it's interesting, exciting. So we started with a cohort of just under 14,000 pregnancies with 12,500 children. And from the start, because of, as well as the interest in gestational diabetes and hypoglycemia and what that was going to mean for the future health of the mother and the child, it meant that we had stored fasting blood samples on the vast majority of the participants. So from the start, we've been in a position where we're able to, by sort of going with begging bowls and working with industry, get, and from the link between Bristol and Bradford and other collaborators, get funds to do multi-omics data, and then eventually to link those data to all of the routine data across the city of Bradford, which means we are already, in terms of the Health Data Research UK Centre, part of Adam Butterworth's multi-omics collaboration, where there are cohorts with multi-omics data and record linkage data, and with a particular emphasis increasingly on diversity in those kinds of settings. So in a nutshell, what we have, I've mentioned just under 14,000 pregnancies, 12,500 women, recruited between 2007 and 2010. 45% are Pakistani, 50% in total South Asian, and the other largest group is the white European population, mostly white British. And I've mentioned there some of the data. We've sort of updated that, in that we've got funding now to get proteomics data on 4,000 of the participants, and are working within the multi-omics consortia to try and add to Bonnie Bradford and other cohorts glycomics data, so there's glycosylation of proteins data. But we also have a massive amount of social and environmental data, green space data, air pollution data, so a huge data set. As with most cohorts, and I think should be the case with all cohorts, these data don't belong to John or me or any particular person. We have custodial sort of governance roles, but the data are there for the research community to use. And any of you who don't know about the study, please have a look at the website and do get in touch about working with the data. We're really excited about that. So I've mentioned the multi-omics data, and one of the concerns I have increasingly in epidemiology is there is so much data around that young epidemiologists, PhD students and postdocs, unlike me, never get the opportunity to collect data. And what I try to do is make sure that they get some insights into how data have been collected, and help then with this promotion of wider use of the data. And this is from the last, what we call Bib Fest that we had in 2019, pre-pandemic, where we bring together the community, the participants and researchers. And these are two of the PhD students that work with me. Kurt, who I believe is on the call, Nancy possibly is. Kurt's a BHF PhD student, and Lucy Pembury from the London School of Hygiene and Tropical Medicine. And they were tasked with bringing together all of the metabolomics data, describing it, and writing a data note that could then be used for others to understand the sorts of data we have in Bonnie Bradford. Okay, so key thing with the study is this linkage to what we call connected Bradford. And we've gone, in fact, from this cohort that lots of people said wouldn't work because Bradford couldn't do something like this, in particular our neighbour University of Leeds, but that's an ongoing Leeds-Bradford thing. And in fact, what's happened is we've extended this with additional cohorts, the Bonnie and Bradford Better Start, which is randomised control trials embedded within a subsequent cohort. But importantly, this connection to all of these data, not just health data, but education and social data. And this is with a really strong link with the local authority. And during the COVID pandemic, which has influenced everything that we talk about today and everybody's lives. Bradford is a vulnerable city. There are pockets of extreme deprivation. There's a large ethnic group. And as everybody did, our first consideration was, how do we make sure our staff are employed and occupied? This dedicated staff of field worker who are going out to schools and out to the community to collect data. And then we move towards how can we work with the local authority primarily. Subsequently, we're writing papers about identifying the most vulnerable areas and using their data and our data to highlight where different inputs were needed. So I'm going to quickly go through some science slides, and then I will talk about how going forward we can use the connection of cohort data with record linkage data on a country level scale, and I guess ultimately globally. So this is work by Tom Bond. The 2021 refers to a preprint on medRxiv. This was recently published in BCM Medicine. And it's asking this question, which was one of many motivations for Born in Bradford, about the link between exposures in pregnancy and future health of the children. And here this is looking at the relationship between maternal body mass index early in pregnancy and body mass index and adiposity in the children later on in life. And what this particular slide shows is a series of results at different ages between maternal pregnancy body mass index and the offspring's body mass index. We saw similar results when we looked at other measures of adiposity, such as skin fold thickness and DEXA scan percent body fat. It combines Born in Bradford data with ALSPAC data, Generation R data, and a Finnish cohort, where within the cohorts we had measures of genetic data on mothers and their offspring, and we had body mass index on the mothers and their offspring at different ages. In each of these age groups, the first result is what we would call the more conventional multivariable regression analysis, where we try to adjust for as many confounders as possible. And then the next set of results are this technique where we use genetic data to try and avoid the problems of confounding. So we use genetic data that relates strongly to the mother's body mass index to examine the role of mother's body mass index without so much confounding. All of the results use a method that we developed for separating mothers and offspring's genetics. So this is the genetic relationship to body mass index that's not through the alleles that are not transmitted to the offspring. And each of these sets of results uses more genetic data in the aim of getting stronger genetic instruments. And in a nutshell here, what we see is in infancy, the observational conventional analysis and what we anticipate being the less confounded genetic analysis are very much in line. So higher mother's body mass index, the higher the adiposity of their child at birth. But as we go through the ages, there's clearer separation, with the genetic analysis tending to attenuate towards the null, though with quite a lot of uncertainty, especially as we get older and we get a smaller amount of data. But how we interpreted these results was suggesting that there may not be a lifelong effect of being exposed to heavier mums in pregnancy, where we know then that there's a greater increase in the metabolic change of pregnancy and of glucose in pregnancy. But that seems to be there in infancy, but doesn't last throughout the whole of life. In analyses like these, and I'll come back to this later, it's very difficult to do differences between the ethnic groups. And there's two reasons for that. One, because this kind of genetic approach is statistically much less efficient than the more conventional analyses. So you're trading off dealing with confounders for statistical power. But the other is that currently, and it is changing and hopefully will change more and more, we don't have much ethnic specific genetic results. Most of the genetic analyses are in largely European studies and get then restricted only to the European populations. So what we have done is some observational evidence in the different ethnic groups, and this is some of Kurt, who I mentioned earlier, the PhD students' work, just looking at ethnic differences in the multi-metabolite data we have in pregnancy on these women, and showing what has been well described, that South Asian women have higher glucose in pregnancy, a sort of higher glucose response to pregnancy than white European women do. But what we hadn't seen described before was that the white European women, in comparison to the South Asian women, have a much more dyslipidemic pregnancy. So a tendency to have a sort of increase in the lipids and cholesterols that are related to atherosclerosis. And that's something we're interested in exploring more. If we think beyond the continuous traits to the different binary thresholds in those teach medical students, doctors need to make things into binaries, yes or no. But for most of those things, there's no magic change at a particular level of glucose or blood pressure, or they're done for a whole range of reasons. And there's variation over time and between countries in where that threshold is used. And this is demonstrated here, the different definitions of gestational diabetes. And at the bottom, what we were doing in this work was seeing what a South Asian specific threshold would look like. And again, this needs further work in terms of, is this the right thing to do? But what's notable is that South Asian women are twice as likely to have gestational diabetes by any of the existing or previous definitions. And if we were to go down the route of an ethnic specific definition, would have 3 times the level. Okay, and then just to summarize this, we have repeat ultrasound scan measures in pregnancy. When we look at that in relation to both glucose and gestational diabetes, the notable things are we see differences in fetal growth emerging in both white European and South Asian prior to that diagnostic time of 24 weeks. So we probably need something earlier to identify gestational diabetes. The pattern of change is identical in South Asian and white Europeans, but South Asian babies are smaller right from the start. And so what that means is that the trajectory of change in fetal growth for a South Asian infant whose mother has gestational diabetes sits exactly on top of a white European woman who does not have diabetes. And then at the bottom here, we showed that differences in fatness at birth based on skin folds and cord leptin levels were between South Asian and white European were largely explained by the differences in glucose. Sorry, I'm rushing a bit because I'm just aware of time rolling on. One of the key things about both working with the community and having the record linkage data was about cousin marriage and causes of congenital anomaly. Very much a political hot potato in Bradford, and the community were keen for us to analyse this but wanted to be involved, and this is work I wasn't involved with. Again, as I said, the study doesn't belong to me or John Wright or Rosie McKechnie. Anybody can use it. This was, I think, really elegant work led by Eamon Sheeran, where they looked at the rates of congenital anomalies by first cousin marriage and by what we know is another risk factor, but is not as used to beat communities up in the same way, older age at which you have a child, the mother's older age. And for both of them, we can see that most of the cousin marriages are in the Pakistani population, though some in other populations. For both, there's a doubling of the risk, the relative risk of having a child with a congenital anomaly for both first cousin marriage and where the mother's age is over 34 years. Important to note is the absolute risk. So again, for both, the vast majority of children are not born with a congenital anomaly. For the first cousins, it goes from a 3% risk if you're not married to a first cousin to a 6% absolute risk. for the age, it goes from a 2% to a 4% risk. And again, sorry that we're running out of time. This is Kurt's work, and this is where he looked at body mass index, alcohol and smoking in relation to congenital heart disease in Braun in Bradford and a number of other studies, all of them highlighting the importance of having record linked data, and found no strong evidence for a relationship between body mass index and congenital heart disease, but some suggestion that smoking may increase the risk of congenital heart disease. And in terms of the record linkage, it's important that that goes beyond antenatal. So in Braun in Bradford, in Alsbach, in many of the studies, we pick up cases through multiple sources, including GP records going up into older ages. So I'm not going to read this out. We've got various plans going forward. What I wanted to do was really sort of finish on this and think about this. So what we have now is we have some sub-studies of various omics and other things within Braun in Bradford. We have the original cohort that were recruited during pregnancy between 2007 and 2010. For some of the mental health and cognitive measures that we did in schools with the sort of mid-child follow-up, the teachers wanted feedback and they wanted the whole class to be done. And that's what was done there. So we have some data then on a larger group. The current follow-up that we're just starting is expanding again to 30,000, where, sorry, that's this one here, where we're going to all of the secondary schools in the whole of the Bradford metropolitan city and inviting all of the children to work with us, and that's already begun. And then we have the whole city of Bradford. And this point here about going from the original 12,500 to, it wouldn't quite be 600,000, but to this large number of the whole population, because it would be, in a sense, the children we're talking about, or the parents. And the bottom bit is going from 2% of the population of the UK to the whole population. And that bit comes from a meeting. I was on MRC strategy board, and it was a meeting in 2015, a sort of away day of everybody. I did a presentation with Sally McIntyre about the future of population health, and there was a big discussion. At one point, John Saville said, 2% of the British population are a member of one of our cohorts. Why are we not linking all of that up to the whole population and using that in our research? And then Paul Elliott was the other population health science person at the meeting, and the three of us were looking at us like this, and I can't remember who, but one of us sort of said, how? And he said, well, I don't know how, but that's for you guys to find out. And in terms of why would we do it now? Because if we do, we can borrow information that I think strengthens all the research. And we've talked about being able to get rare disease outcomes. It's important if we're interested in ethnic differences that we can increase the power in some way. But there's also emerging work from people like Ruth Keogh that suggest when you've got nested studies, even if they're a relatively small proportion, there are advantages in imputing the data up to a higher level. It seems incredible. It's the sort of thing I can remember when we started first doing imputation, George Davis Smith saying, you mean you're making up data? So, you know, it means making up data potentially for millions. But it's not necessarily going to increase power. It may in some circumstances get better confounder adjustment. And we can also use the bigger data to inform sensitivity analysis and inform what we might expect to be happening, and go from the smaller data out to the bigger data. It's part, my BHF chair is linked to my IU programme, and as we're doing the renewal for that with Kate Tilling and a lot of smart collaborators, we're interested in the extent to which you can go with this and how we can best borrow from these different layers of data. And apologies, I think I've run over time, but thank you very much for listening.
Deborah, thank you very much. That was an absolute tour de force. Thank you very much. And you couldn't have finished on a better note to pick up a question. I think we've got time for one. So just to really kind of bring you right up into feasibility and practicality space based on everything you've said and what you were just saying now, the next step is to recruit 5 million people into the Our Future Health initiative. So based on your experience with Born in Bradford, what would be your advice around recruiting a representative, inclusive and ethnically diverse cohort at that kind of scale to that team?
Yeah, and it's something that's been discussed for a long time and was discussed when I was involved in the MRC for a long time. I think it's really tricky, the balance between representativeness and the alternative of going to specific areas and regions that try to capture diversity, but also I think that support that idea of being embedded within the city and being able, it's perhaps difficult to, or maybe you can learn from, I mean, Scotland have done it, we've done it in Bradford. It's been very much about working with the community, bringing the community together, not making assumptions. I think we did some things well, we did some things badly to begin with. We were sort of almost reaching out to the community separately. So going to areas where predominantly the population are South Asian, going to the local mosques, and then going to some of the more deprived white British areas, going to the football clubs, going to these different things. And it worked to a certain extent, but what kept coming back was, why are you not bringing us all together? Why are you not getting us all into the room? Which is what we do now with the sort of annual Bristfest. And we go to the different schools. I think you get more diverse populations there. But it is about having, a bit like Kelvin, but other people telling their stories about how the use of their data is making a difference, how they want to be involved with how the data is used, and doing it in ways that don't, I guess, feed the fire of some of the tensions that there are in different communities. But I think statistically we can learn, just going bigger, is what we plan to do in Born in Bradford, because we have all these data connected up, is push and try these different statistical methods that I then think can inform bigger cohorts and bigger things like our future health.
That's fantastic. Thank you very much. Really great advice. And I think this need for engaging, involving, not just informing, but actually finding the stories from our diverse public and publics is going to be a recurrent theme throughout the rest of the day. So thank you very much to everyone from that previous session, and in particular to Deborah for her fantastic keynote talk. We now have a break for lunch, but if you want to hear about imaging, trials, wearables, phenotyping, and what the BHF Data Science Centre is doing to support cohorts and their challenges, then please do come back after lunch. We'll be kicking off again at 1:15, and have an enjoyable break in the meantime. See you all again soon.